Hello, welcome to the Muscle Lab of Fall 2020. This lab, or this video from this lab, will correlate with pages 85 to 88 in your third edition lab manual. But in order to really understand this material that I'm going to present to you, you really have to go into chapter 9 of your textbook, Pearson Marriott, and start learning some of these words. Now I'm going to skip to page 87 to start here to just give you the packaging of these muscles, right? Because you're going to start looking at muscles. You already know that muscles move bones, muscles move joints. We're going to talk a little bit about the wrapping of a muscle, get that done. You know, a tendon connects muscle to bone, or bone to muscle. A ligament will connect bone to bone. But here we're talking about the packaging of muscles. So you have to picture a tendon which begins with dense, regular connective tissue, not very vascular. So if you ordered a muscle on Amazon, it would come packaged, right? The outside would be a good protective package. So in a muscle, the tissue, which is an extension of the tendon, we call this fascia. So sometimes we say fascia connects muscle to muscle. So the outside of a large muscle is called epimysium. Epi meaning the top, the outside. So the complete outside layer. Then there's another wrapping as you go inside a muscle, and you're going to get these areas called fascicles, and they're covered by perimysium. So the fascicles is just another descending layer of a muscle. So you should be able to identify a muscle. You should be able to go and see a muscle and see that this is the brachioradialis, right? Or the extensor digitorum or the deltoid. That's the full-sized muscle. So the wrapping begins on the outside of that muscle with the connective tissue, fascia, continuous of the tendon called epimysium, and then smaller to perimysium around the fascicles. And then within the fascicles, you're going to have the actual muscle fiber. And the muscle fiber is just another name for muscle cell. We're talking about skeletal muscle here. So this is voluntary muscle that we're talking about. So within the muscle fibers, you're going to have what's called myofibrils. And that's going to be surrounded by another inner connective tissue called endomysium. Mysium is muscle. And it's connective tissue here, right? So the tendon, the epimysium, the perimysium, the endomysium, and you should be able to descendo that down into the muscle fiber, into the myofilaments. And the myofilaments are within the myofibrils, which were within the muscle fiber. So the myofilaments is where we're going to live. That's the microscopic anatomy that we need to talk about. You're going to hear terms like sarcolemma. Sarcolemma is the membrane of the muscle cell, or the muscle fiber, or the myocyte. And that's the exciting place. It's an excitable membrane. Just like nerve tissue, it's an excitable membrane. So there's going to be things like depolarization, action potential, repolarization. Very important for muscle contraction, because a muscle, really all it does is shorten to do work, and that's contraction. Right? So we'll learn about this other smallest unit of a muscle fiber, and that's called the sarcomere. So we're going to take a look at that on page 85, and that's the sarcomere. So get these coverings down, not as difficult as learning the sarcomere. So what's going to happen at the sarcomere, and I go to page 85, and you're going to hear terms. You're going to hear about proteins. You're going to hear about contractile proteins, regulatory proteins, and structural proteins. Okay? So if you look at page 85, you see these lines where you have to fill it. Now, this should help you. This video, the PowerPoint that the TAs and I will post on Blackboard, and your book will help you with this. There's going to be a lot of new words that you really never heard before, possibly. So the sarcomere, again, is the smallest unit of the muscle. And we use sarco because sarco means muscle, right? So instead of plasma membrane, we call it the sarcolemma. Instead of the endoplasmic reticulum, we call it the sarcoplasmic reticulum. Instead of the cytoplasm, we call it the sarcoplasm. So that's really important that you get those terms down, going down. So you're going to hear of proteins. And there's the sliding filament theory, which is going to involve contractile proteins. And I'll give you the names. The contractile proteins are actin, which is a light fiber or a thinner fiber. And then we have myosin, which is a thicker fiber or darker fiber. So they're the contractile proteins. They're going to make the sarcomere shorter, okay? And when you're learning muscle, you're going to learn about an important thing. First is this action potential from the nervous system, from the motor neurons, from the axon of the motor neuron. So that has to excite the cell membrane of the muscle to depolarize, to get an action potential. And then a lot of things happen. One of the main things that happens is the release of calcium. Calcium is important. You learned about that in bone. You learned about calcium homeostasis. This is why it's important. Your body really doesn't care about your bones as much as it does the blood calcium for muscle contraction, nerve conduction. So, first of all, that has to happen. You have to have an action potential. You have to have calcium release, which you're going to learn. Calcium is stored in the sarcoplasmic reticulum. This is not a place where we're really concerned about building proteins and steroids and phospholipids. In the muscle fiber, we're really interested in the storage of calcium and the release of calcium. So when we need a muscle contraction, that sarcoplasmic reticulum is going to release calcium. You know calcium, the ionic calcium, right? And if it wants to stop a muscle contraction, when the muscle needs to rest or go back to its resting state to stop a muscle contraction, that calcium is going to be sequestered back into the sarcoplasmic reticulum, right? So actin and myosin are the contractile proteins. Then you have two other proteins that form a complex. They're called troponin and tropomyosin. So troponin is a little bit smaller. Tropomyosin is a little bit longer, which you could see in this diagram. You can see it on the PowerPoint. You'll see it in the book. So know this. Calcium has to bind to troponin in order to remove the tropomyosin from actin. So tropomyosin in the resting state is blocking the binding sites for myosin on actin. Okay? So what calcium does, when calcium is released from the sarcoplasmic reticulum, it's going to bind to troponin. That complex, troponin and tropomyosin, is going to move away from the binding sites for myosin on actin. So tropomyosin is covering the binding sites. So once the calcium binds, the tropomyosin will be displaced. And now the myosin could bind to actin. So this sarcomere, which you could see on page 85, in the relaxed and the contracted state. So there's an optimal length you need for these sarcomeres in order to get a good contraction, in order to get a good binding of myosin to actin. So if you look at the outside, you have this big red part on the ends, the two red or pinkish areas. And those are going to be called the Z disc, right? Z disc, they're thick. Okay, so anatomically, a sarcomere is defined as Z disc to Z disc. Okay, so everything in between, as you can see. So what's going to happen, and I told you, or I may mention, that really all the muscle does is shorten to contract. So when all of those sarcomeres and all those Z discs in the sarcomeres all come together, they're going to shorten to do that work of the muscle. So anatomically, that's Z disc to Z disc. So the middle, the dead center, if you look at the dead center of a relaxed muscle, that's going to be called the M line. And the area where you see the arrows on top, the two arrows pointing towards the outside, that's called the H zone. So if you look at the bottom, the contracted muscle, you're going to see that the Z discs have come together and the H zone basically disappears in a contracted muscle, maybe in a fully contracted muscle, right? There's degrees of contraction, okay? So there's another protein called titin, and that's mostly for structure, strength, integrity. There's other proteins you might hear of a nebulin or herculin or dystrophin. So they're all about the integrity of the sarcomere, what gives it strength, okay? So if you look at the actin and myosin, you look at this diagram, and they're relaxed first, you're going to see that the myosin looks kind of like a string of beads. That's a protein. It's a polypeptide chain. So it's a little thinner and it's a little lighter. And the binding sites for myosin are on actin. But in the resting muscle, you're going to have tropomyosin blocking those binding sites. So actin is kind of a string of beads, if you want to describe it in that picture. It's blue in that picture. Now, the myosin is more shaped like a golf club, which has a head or a hurling stick or a field hockey stick, right? So the head of that. So the head of myosin is going to bind to actin onto those binding sites that are covered with tropomyosin in the resting muscle. And what myosin is going to do once it connects is swing that actin towards the center of the sarcomere. And this happens on both sides because you notice there's two Z discs. Okay? And you might have to label what's called an A band and a I band. And that's within the sarcomere if you look at the histology, the study of this tissue microscopically. So I always remember where you have the thickest filaments, proteins like myosin, that's going to be dark. And there's an A in dark, so I like to call that the A band. And there's A band on both sides of the H zone. So the A band is thicker. You might hear of the term striated. So these proteins, the darkness of these proteins give it that striation, the term striation. And you see that in cardiac muscle as well. Now the I band is more on the periphery. So the Z disc is basically right in the middle of an I band. An I band, I remember light fibers, because light has an I in it, I band. So dark is the A band. I band is the light area when you look at the histology. So all of these filaments of actin are going to swing to the center of the sarcomere. So that's really important. So this is what happens. And you're not learning the neuromuscular junction until next semester. We talk about the neurotransmitters. You should be aware that there has to be an excitable membrane. There has to be an action potential. Because you got to release that calcium from the sarcoplasmic reticulum. If that doesn't happen, you're not going to have a contraction, right? Okay, so and this is the muscle cell. So once the calcium is released from the sarcoplasmic reticulum, it's going to bind to troponin. Tropomyosin complex is going to be moved. So the tropomyosin is going to move away from the binding sites on actin for myosin. So now the myosin heads could bind to actin. And then it can move and swing the actin towards the center of the sarcomere, which is very important. So we need energy for this. This is why muscle cells have so much mitochondria. Mitochondria, through aerobic respiration, using oxygen. So we need oxygen for aerobic respiration. That's what it is. And you're making excess amounts of ATP because muscle contraction needs a lot of ATP, right? Because that's the energy that's going to actually get this swinging of the actin towards the center of the sarcomere, all the way through the whole muscle, right? So once myosin could bind, well, once the calcium binds to troponin and the tropomyosin is displaced, we got to go through a few steps. And on page 86, I'm going to simplify these steps for you. First of all, you need the calcium. Then you need the binding of calcium to troponin. Then you need tropomyosin to be displaced off the binding sites on actin for myosin. Then we got to energize the head of myosin to attach. So listen to this. The first step of this sliding filament theory is we have to hydrolyze ATP. to get energy to energize the myosin head. So that's really important. Hydrolyzing is when you take a bigger molecule like ATP, it's adenosine triphosphate, and we use water to break it up, right? Remember, the opposite was dehydration synthesis. So we have to hydrolyze the ATP to get that powerful inorganic phosphate to energize the myosin head. So that's the first step. Hydrolyzing ATP. That's step one of the sliding filament theory. It's a good theory, right? And then the second step is going to be the binding of myosin to actin. I like to call that cross bridge. And this is happening at every molecular level in the sarcomere with these proteins. So step one is hydrolysis of ATP to energize the myosin head. Step two is the binding of myosin to actin. I call that a cross bridge, right? The third step is using that energy built up to bring that actin towards the center of the sarcomere. Step three, I like to call that the power stroke, bringing the Z discs closer together with that actin swinging to the middle. So it's power stroke, like, Eddie Van Halen power stroke, big time thing, right? So step one, hydrolysis of ATP to energize the myosin head. Step two, the cross bridge, the binding of myosin to actin. Step three is the power stroke, the swinging of the actin to the middle of the sarcomere, which is towards the end line, if you like, and bringing the Z discs closer together, shortening the sarcomere, shortening the muscle. We could do work. So the last step of muscle contraction is basically detaching that myosin from the actin head, and that needs energy. We're going to need ATP for that. So you need more ATP on top of that. So the last step is detachment. First step, hydrolysis, energizing the myosin head. Second step is your cross bridge formation. Third step is your power stroke. Fourth step is detachment. You need ATP. Like if you're a dead guy, you have no oxygen, right? Shortness of breath, you have no oxygen. So if you have no oxygen, you make very little ATP. So, if you watch CSI and those kind of shows, there's a condition called rigor mortis, where the muscles are still attached for like 12 to 24 hours. And because there's no ATP, because you don't have any oxygen. So they can tell the time of death based on that condition called the rigor tightness mortis death, right? So rigor mortis is based on not having ATP to detach that muscle. So we want a muscle contraction to stop. Of course, it's much more complicated than that. You have other muscles. You have muscles that fuse tetanus and more postural muscles. But we're talking about a single contraction of a muscle, shortening of a sarcomere. Okay? So here's the players. You have your calcium. You have your sarcolemma. You have your sarcoplasmic reticulum. You have troponin, tropomyosin, actin, myosin, titin, Z discs, A bands, I bands, and of course oxygen and ATP. Now on page 88, you have kind of breaking down the classification of muscles, of course. So I think it's important that when you're learning muscles, first you have to be able to identify it, and you're going to do that, during these labs. And you're going to see what muscle is what, where they are, what group they're in different areas of the body. So you're going to have to name them. That's a nomenclature that you're going to have to start learning and what it means. Try to put it together with a meaning. Shape is rhomboid or deltoid or trapezoid, trapezius. Then I think the next thing you should do is see what the action is. Like the biceps brachii flexes the elbow. And then maybe you can figure out the origin and insertion, because the origin is usually a fixed point. And the insertion is usually the moving bone. So in my biceps brachii, when I flex my elbow, really my forearm is moving, or my radius bone is moving towards my shoulder, becoming more proximal. So learn the muscle, the action, the origin insertion, which is quite difficult to memorize a lot of these, but if you think of it that way, it makes more sense. Like, you know what the pollex is now, you know what abductor, abduction is, abductor pollicus, longus, or the extensor, extending the thumb, extensor pollicis brevis. Or extensor digitorum is talking about extending the forearm. The fingers, really, I'm sorry, the fingers, because digits. And then there's carpi, which is wrist. Carpals are your wrists. So you should remember that. Always remember in the anatomical position that the radius is on the outside, or lateral in the anatomical position. And the ulna is medial. Same with the fibula in the lower leg. Fibula is always lateral. Tibia is medial. So it all goes back to the beginning. We talked about tissue here. We talked about proteins, connective tissue, nerve tissue. All the tissue is involved when you get to this point. And now we're talking about muscle tissue in a major way. So muscle is really a working tissue, right? Skeletal muscle is voluntary. Cardiac muscle, you learn later, is involuntary, thank goodness. But and with metabolism, like we talked about ATP, oxygen, and of course we're going to need macromolecules like glucose to create that energy, either anaerobically or aerobically. So skeletal muscle, if I asked you to go run 1,600 meters out of the track out there, you could run and you'd be real tired or walk, but you could pay that energy back. You can pay that oxygen debt back, right? No problem. You'll be running in a few hours or the next day if you're not too sore. But cardiac muscle is a little different. Cardiac muscle, you need to contract that muscle from the time your heart starts beating till the time it stops. So that means ATP flow constantly. And it's very similar, just that one's voluntary, one's involuntary. You learn about cardio. So your skeletal muscles are basically like using a credit card. You buy now, pay later. Heart, cash only. ATP, oxygen as you go. So the metabolism is very important. What happens inside that sarcomere is what we're interested in this video and in lecture 2 when you get to that. Okay, so on page 88, talking about a little Shakespearean situation here with agonist versus antagonist. So the agonist is the one muscle or the group of muscles that's producing that movement. You should know the movements. You should know flexion, extension, abduction, adduction, circumduction, eversion, inversion, pronation, supination, lateral flexion, protraction, retraction, all of those movements. Okay, and which one is the agonist? Which one's doing it? Like if I bend my knee, or if I flex my knee, my hamstring is doing the work. So the hamstring is the agonist. I extend my knee upward, and the quadriceps are the agonist. So remember the quadriceps are hamstrings. Hamstrings are the posterior thigh muscles that flex the knee. So when the knee is flexing, like if you step on a nail, your hamstrings are the agonist and your quadriceps are the antagonist. So you learn about that. Sometimes we use what's called a synergist, another muscle that helps control or assist. It's recruited to help that movement. So synergy means to help out, to add something to what's going on there. So this is really important as far as how muscle contracts. Again, skeletal muscle is voluntary. We use a place in our brain, that thing in your cranium, your cerebrum, your cerebral cortex. And that's really where the action potential starts. I really don't know how, but you have an action potential when you think about bending your knee or running 1,600 meters. Okay? But you need balance. You need coordination, and your brain has a lot to do with that too. That may be subconscious, unconscious, but we're learning voluntary movement, specific muscles. Know the actions, know the origins and insertions, and you'll get to know the nerves that control. Because all muscle needs a nervous impulse, action potential from a nerve. You might have an argument with the heart, but the heart is controlled by the nervous system, not this nervous system. So the nervous system that moves voluntary muscle, the part of our nervous system that we use volition, we could think about moving, we could cognate what we're moving. We call that somatic muscle contraction. And we have reflexes too, which you don't need your cerebral cortex for reflexes, which you'll learn. But those are skeletal muscle somatic reflexes, as opposed to autonomic reflexes, which are more controlled by deep in your brain. You're going to learn that when you do the nervous system. So remember, we're talking about motor muscle contraction, somatic muscle contraction, voluntary muscle contraction of skeletal muscle. You know the other two. You know smooth Talk about them when we get to blood vessels and digestive and reproductive next semester. Reproductive. When a muscle immobilizes a bone, when it stops it from moving, it's called a fixator. That's an interesting one. So the way these fascicles, remember the fascicles, which are made up of the muscle fibers, and the muscle fibers have the myofibrils within them, and the myofilaments are your actin myosin filament. But the fascicles are arranged differently. Like you might have heard of a muscle called sphincter, which is a round circular shaped muscle, and that's the way it contracts, where the fascicles are in a circular movement. So we do have skeletal muscle sphincters. Not all of them are. Some are smooth muscle, but the ones we can control. When you do urinary, you'll see that outside the towards the urethra, and in the digestive system towards the anus, you have voluntary circular skeletal muscle structures. But a lot you're going to learn about blood vessel sphincters and your sphincters in your digestive system, pyloric and cardiac, from your stomach, or mostly smooth muscle. So we have these different types of muscle here, and you can read the bottom. It tells you different types of fusiform, multipennate, and you should be able to name those muscles based on that model. It's very well done. So a convergent muscle is like the pec major, or the spinatus, where the muscle converges from a large arc to a small tendon and inserts, in this case, on the humerus. Then we have fusiform and non-fusiform. So the biceps brachii, if you look at it, it's kind of like a thick belly in the middle, and fusiform shape, meaning the two ends, the origin and insertion tendons, are smaller and the belly is thicker. So that's a fusiform. A non-fusiform would be this, I don't know, see if it has it, is this really long. This is the longest muscle in the body, the sartorius. Looks like a tailor's measuring tape. So they call it the sartorial, right? Sartorius. So that is a non-fusiform parallel. And then the bipennate would be this one, coming in two different oblique. You'll see this when you come into lab. Hopefully you'll come into the lab. You'll see it close up on the PowerPoint or in your book. You have two oblique fascicular directions coming down to this one tendon. Remember, tendon connects muscle to bone, ligament connects bone to bone. So above the patella, you have a tendon. It's the quadriceps tendon. So below the patella, you have the connection between the patella and the tibia, and that's the patellar ligament. And that's the one the doctor hits you on when he whacks you with the hammer to check your reflexes, skeletal muscle reflex, somatic reflex. But a reflex, you don't need your cerebral cortex. It's just checking those particular nerve roots. So multipennate would be your deltoid. Many different oblique fascicles coming towards your common tendon. Remember the deltoid tuberosity? That's going to be your insertion. And you're going to get some abduction from the deltoid, away from the midline. Okay? So go to your book, start doing the worksheets in here, and use the PowerPoint. I'm sure I'm going to put it on mine, and the TAs will have it, and it's really helpful if you don't have time to get to the book before that. But please read this chapter. There's a lot of new words here. Okay? Good luck.